Hi, so in this video I'm going to talk about the molecular mechanism that governs lytic and the lysogenic cycle. This video is divided into several parts, so make sure you watch till the end. Before we begin, let's start with the difficult part where most of the students gets confused. And I want you to remember this. PR will cause transcription of CRO and C2. PRE will cause transcription of CI repressor. Let me repeat this. CRO and C2 are under the control of promoter PR and CI is under the control of promoter PRE. Now besides PRE, there is also a promoter called PRM. PRM will also cause transcription of CI. So just remember this. Now let's try to understand the map of phage genome and the function of all these genes. On the top, we have the gene CI. CI codes for a repressor protein. Then we have two strong promoters PL and PR. Here L means left and R means right. On the right side, we have several genes like CRO, C2, genes for DNA replication and cell lysis. Then on the left, we have genes like N, which is anti-terminator, C3 and genes for recombination. Now let's see step by step what happens when the lambda phage genome enters the host cell. Once inside the host, the two strong promoters PL and PR will begin the transcription of gene N and CRO. At the end of these genes, there are termination sequences, so the RNA polymerase will stop transcription. Once the proteins are translated from the mRNA, the N protein will act as anti-terminator. The N protein binds with the RNA polymerase and modifies it. This modification allows the RNA polymerase to ignore the termination sequences and proceed further. This finally allows RNA polymerase to transcribe genes like C2, C3 and genes for DNA replication. Now C2 can be easily degraded by the proteases found in the cell. This degradation is prevented by C3. C3 inhibits the host protease enzyme. As the protease is inhibited, the degradation of C2 is prevented. C2 now binds the C2 binding site. After C2 binds with the C2 binding site, the transcription from PRE begins. This will lead to formation of CI. CI is a repressor. Now let me take a pause and introduce you to two more terms, PRE and PRM. PRE stands for promoter for repressor establishment. PRM stands for promoter for repressor maintenance. The CI repressor binds OR1 and inhibits the promoter PR. This binding will eventually inhibit formation of CRO, C2 and genes for lytic cycle. As the genes for lytic cycle cannot be transcribed, the phage genome finally undergoes the lysogenic cycle. Now comes the important part. This is where the CIII repressor becomes cunning. The strategic binding of CIII repressor on OR1 keeps OR2 and OR3 free. OR2 and OR3 are the part of promoter PRM. The PRM promoter allows continuous transcription of CI. As a result, there is more and more formation of CI. Now let's see the working of CRO. CRO binds OR3, which is part of PRM. This binding inhibits the promoter PRM. As PRM is inhibited, the transcription of CI stops. So, CRO inhibits CI formation. At the same time, look carefully. OR1, which is part of promoter PR, is free. This allows transcription of all the genes on the right side, including CRO. As the genes for replication, head and tail are expressed, the phage undergoes lytic cycle. Now let's talk about the competition between CI and CRO. So this is what we have seen so far. CI binds OR1, whereas CRO binds OR3. So the question is, who wins this competition? The answer to this question depends on several factors. The first one is the amount of nutrients available to the host. If the nutrient concentration is high, the host is metabolically active. As a result, the host will have large number of protease to degrade C2. If C2 is degraded, CI is not formed. Now, if CI is not formed, then CRO wins the competition. As the genes for replication, head and tail are activated, the phage undergoes lytic cycle. Now let's see the scenario where the amount of nutrients available to the host is less. If the nutrients are less, the metabolism will be slow. As the metabolism is slow, there will be less amount of protease present. Since the amount of protease is less, C2 survives the proteolytic degradation. C2 will allow formation of CI. CI then inhibits OR1 and inhibits the genes for lytic cycle. As a result, the phage undergoes lysogenic cycle. Now let's see the second factor that governs the competition between CI and CRO. This factor is DNA damage. When the DNA is heavily damaged, the SOS repair gets activated. During SOS repair, we have active REC-A. REC-A degrades CI. As a result, CRO wins and it favors the lytic cycle. Okay, now let's see the third factor that governs the competition between CRO and CI. And this factor is the number of phage genome entering the host cell. If the number of phage genome entering the host cell is more, then more amount of C2 is produced. As C2 is more, CI will survive and it will promote the lysogenic cycle.